Hey everyone, this is the lab safety briefing that I gave Group A on Thursday, I believe. I just wanted to apologize again for missing out on some of you with Group B. This is kind of new for me too, having to split everyone up into sections, and I got distracted by other work and just didn't even bother coming. So this will make up for that. Fortunately, it wasn't too much. He didn't miss anything of real, academic value. It's important with regard to safety, but we'll get you that caught up with that all right here. So I want to give you kind of an overview of the lab since in case you haven't been in the biology lab before. So this is the door that you'll be coming in. So when you come in, you'll see the lab and you'll just sit at one of these benches. Remember to maintain social distancing. There's usually 2 per bench is sufficient. I think there's 12 or 13 in each section, so there'll be 2 at pretty much every lab bench. With regard to safety though, I'm just going to go over that safety brief, which is on the board there. First thing in this lab that you'll notice on the door when you come in, there is no food or drink allowed in this lab. Now for the most part, the classrooms in Cloud, I think, allow them, but the YL 224, the biology lab, does not allow it. And the reason is that this is where I teach my microbiology course. And so with regard to microbes, my students work with these. My micro students work with these a lot. And you'll see these are all sorts of nasty bacteria growing on an agar plate. They tend to do a good job cleaning up after themselves, but not always. And so if there's on the bench a bunch of bacteria that was spilled and you bring a food or drink in, rub it all over your fingers, rub it all over your food, and then eat it, it's possible that you could get an infectious dose of a bacteria. Most bacteria that we work with are not infectious in this laboratory, so it's not a big deal, but I just want you to be aware that sometimes we do work with BSL-2 or biological safety level 2 organisms like E. coli or Staph aureus or Mycobacterium. And so there's a few bacteria that you wouldn't want to get a real high dose of. So that's why no food or drink. If you do have a drink, make it a closed drink and keep it in your bag. So if you need to get a drink during class, I know these masks are really annoying. I haven't shaved very much because of the dang masks. So that's one benefit, I guess. But if you do need a drink, like I said, keep it in your bag, have it a closed bottle so you can just open it and drink as you need. The next thing, when we are working in the lab, you'll notice that I tend to stockpile all the equipment, let's go back here, on this bench right here, this table. And so right here you see a dissection tray, but normally all the beakers and grad cylinders and all the liquids that you need will be over here. And so as you set up your lab apparatus, if we have lab apparatus to set up, that you not push, trip each other accidentally, be polite in the laboratory. There's plenty of time. Most of my labs take half the time, if that, so you should have no issues completing things in an orderly manner. With regard to wear in the lab, number 3, I worked in a pharmaceutical lab for a number of years, and so yes, I had to wear long pants, lab coat, and business casual when I was in the lab. We don't work with anything that dangerous in the laboratory, so you can wear whatever you wear to school. If you're wearing, it depends on how early the lab. Some of my general biology students will come like right out of bed. They'll have like tank tops and pajama pants on. I'm totally fine with that. Just be aware, if you wear something nice, sometimes we work with stains, microbial stains, and if you get that on your clothes, you're going to ruin your clothes. So dress appropriately, but if you're careful in the lab, you won't have to worry about getting any stains on your clothes. We don't, as I mentioned, I don't use a lot of noxious chemicals in here, so I don't require goggles, but if you like goggles or to wear safety shield glasses, that's fine. I can provide them, you can bring your own, either way. Whenever we're doing dissection or doing staining, I will require gloves, and so there's gloves up here on my bench, and there's gloves in the back, right around there, and those are for your use when you're in the lab. If you do get microbial stains on your hands and you don't have gloves on, you're going to be purple or you're going to be red until that skin dies and falls off. So it behooves you to wear gloves when I tell you that you should be wearing gloves. If you are allergic to anything, if you have chemical sensitivity allergies or asthma, or if you're a female and you're pregnant, it's important that you tell me this. Not necessarily because anything we do is dangerous to a fetus or could trigger an allergic episode or an asthmatic episode, but I don't want you to be uncomfortable. So if you're pregnant and you're worried about your fetus and you don't want to be around the noxious fumes, we can work something out. With regard to hair, it doesn't matter what sex you are. If you have long hair, it's best to either keep it in a hat, keep it in a bun, keep it in a ponytail. You need to keep it out of your face, basically, because if we're working with a stain on the table, and you'll have a tray here, and I've had females before with their long hair, they'll be down working, and they'll turn their hair, and their hair will fall into the tray. So then they have purple or red streaks in their hair. The glassware that we use, beakers, grad cylinders are all glass. It's okay if you break one. Don't try to break it, but if you do, just let me know. I'm not going to bill you. I'm not going to yell at you, but we do need to take care of it. And so in the back, I'm in the back of the lab, so there's the front. If I just turn around here, you'll see on the floor there's a glass, there we go, a glass waste. So you heard that there's a lot of glass in there. It's because students break things. Again, no big deal. If you do break something, though, use that hand broom and clean it up. Media with microorganisms. So like this one that I showed you before. I think we will use one plate at least in this class. If you have media with microorganisms, this does not go in the trash. So I'm again in the back of the class, spin around to give you some reference. So we're going to walk back into the back room here that is usually dark. And back here is the autoclave. And so you'll see on this bench here, there's an area with a bunch of plates that says needs autoclaved. You can't read that. And so all this media will have to be put inside this machine. And it gets very hot at a very high pressure. And that's so it'll kill any microbial spores. And so inside this autoclave, you can see she has biohazard right now. And so that stuff has been sterilized and is now able to be thrown away. So we don't ever throw away microbial media like agar plates, agar slants, media broth. We tend to autoclave that first before we discard it. Chemical waste can go in the sink. The sink is in the side of the room here. You'll notice there's no handles. It's because it's a surgical sink. So you have to stick your leg in here, try to get that for you, and pull it. And so you're able to, with dirty hands, turn it on and off just with your leg. Now you may hear that when I turned it on, the water started coming out pretty darn fast. And so if you're trying to clean out a tray full of stain, if you put your stain in here and then you turn it on full blast, that's going to spray up and hit you. I've had students do this. They'll get red and purple polka dots all over their shirts because they blew that stain all over their shirts. I've also had students blow it all over their face because it'll hit the sink and splash up into their face, and then they have little red and purple polka dots on their face. So just be careful when you're working with those microbial stains. Those of you that are with child or have children, I have 3 myself. I understand it can be difficult to go to school with children. I went to pharmacy school with 3 children, so I understand. But with regard to lecture, if you had to bring a kid to class, I'm totally cool with that, as long as they're quiet and just sit there and don't disturb us. In the laboratory, I can't do that, though, because you'll notice there are all sorts of pieces of equipment. We'll be working with dead animals, not a lot, but when we dissect worms, ascaris. If you take Bio 2, we're dissecting a cat. We're working with scalpels, some chemicals. I just can't have little kids running around. So unfortunately, if you can't find childcare, you're just going to have to miss class that day, I guess. And then finally, when you are working with glassware, which we will, we need to make sure we clean it after we're done using it. And so again, in the sink, you will just clean and rinse it off. On that same tray or that same cart with all the equipment, you will just put a paper towel, and there's usually paper towels over there. Do that one-handed. Paper towel, rinse off your beaker and put it upside down on top of the paper so it can dry, and then the next class period can use it. It's important that you know the location of the eye wash station and the fire extinguishers. Fire extinguishers are by the front door where you come in. You'll see it in the corner there. Eye wash station is in the other corner, and hopefully you can see that big yellow sink there. If you need to use the eye wash station, which we have had students need it, you just push this back. And when you push it back all the way, there's a pressure that'll blow these caps off. But as you can see, there it goes. It's perfectly fine if you need to rinse your eyes out because you get maybe formaldehyde in your eye or you think you got some dye in your eye, to use that. The body wash station is the big shower looking thing, and you just pull this lever down and it'll gush water out of this thing up here. You'll notice there is no floor sink. So fortunately we've never had to use this. If we did, we'd have to close the lab down because there'd be just a crap ton of water all over the floor. I think we have to call in a hazmat kind of team to come in and take care of it. So please don't pull that unless it's absolutely necessary. And I cannot think of a single situation where it would even be close to necessary in the Principles of Biology lab. So that takes care of the lab safety. So again, go finish the lab safety acknowledgement or biosafety acknowledgement, whatever it's called. I think you just have to type I agree, or just do what the assignment says and get the 10 free points. So I'll talk to you again soon. If you ever have questions or ever need anything from me, please shoot me an email. Have a great one.